Special techniques. Reversed phase chromatography. Reversed phase chromatography is any liquid chromatography procedure in which the mobile phase is significantly more polar than the stationary phase. It is so named because in normal phase liquid chromatography, the mobile phase is significantly less polar than the stationary phase. Hydrophobic molecules in the mobile phase tend to adsorb to the relatively hydrophobic stationary phase. Hydrophilic molecules in the mobile phase will tend to elute first. Separating columns typically comprise a C8 or C18 carbon chain bonded to a silica particle substrate. Hydrophobic interaction chromatography. Hydrophobic interaction chromatography is a purification and analytical technique that separates analytes, such as proteins, based on hydrophobic interactions between that analyte and the chromatographic matrix. It can provide a non-denaturing orthogonal approach to reversed phase separation, preserving native structures and potentially protein activity. In hydrophobic interaction chromatography, the matrix material is lightly substituted with hydrophobic groups. These groups can range from methyl, ethyl, propyl, butyl, octyl, or phenyl groups. At high salt concentrations, nonpolar side chains on the surface on proteins interact with the hydrophobic groups. That is, both types of groups are excluded by the polar solvent. Thus, the sample is applied to the column in a buffer which is highly polar, which drives an association of hydrophobic patches on the analyte with the stationary phase. The eluent is typically an aqueous buffer with decreasing salt concentrations, increasing concentrations of detergent, or changes in pH. Of critical importance is the type of salt used, with more kosmotropic salts as defined by the Hofmeister series providing the most water structuring around the molecule and resulting hydrophobic pressure. Ammonium sulfate is frequently used for this purpose. The addition of organic solvents or other less polar constituents may assist in improving resolution. In general, hydrophobic interaction chromatography is advantageous if the sample is sensitive to pH change or harsh solvents typically used in other types of chromatography but not high salt concentrations. Commonly, it is the amount of salt in the buffer which is varied. In 2012, Muller and Franzrib described the effects of temperature on HIC using bovine serum albumin with 4 different types of hydrophobic resin. The study altered temperature as to affect the binding affinity of BSA onto the matrix. It was concluded that cycling temperature from 40 to 10 degrees Celsius would not be adequate to effectively wash all BSA from the matrix but could be very effective if the column would only be used a few times. Using temperature to affect change allows labs to cut costs on buying salt and saves money. If high salt concentrations along with temperature fluctuations want to be avoided, one can use a more hydrophobic to compete with one's sample to elute it. This so-called salt-independent method of HIC showed a direct isolation of human immunoglobulin G from serum with satisfactory yield and used beta-cyclodextrin as a competitor to displace Ig from the matrix. This largely opens up the possibility of using HIC with samples which are salt-sensitive, as we know high salt concentrations precipitate proteins. Hydrodynamic chromatography. Hydrodynamic chromatography is derived from the observed phenomenon that large droplets move faster than small ones. In a column, this happens because the center of mass of larger droplets is prevented from being as close to the sides of the column as smaller droplets because of their larger overall size. Larger droplets will elute first from the middle of the column while smaller droplets stick to the sides of the column and elute last. This form of chromatography is useful for separating analytes by molar mass, size, shape, and structure when used in conjunction with light scattering detectors, viscometers, and refractometers. The 2 main types of HDC are open tube and packed column. Open tube offers rapid separation times for small particles, whereas packed column HDC can increase resolution and is better suited for particles with an average molecular mass larger than Daltons. HDC differs from other types of chromatography because the separation only takes place in the interstitial volume, which is the volume surrounding and in between particles in a packed column. HDC shares the same order of elution as size exclusion chromatography, but the 2 processes still vary in many ways. In a study comparing the 2 types of separation, Eisenberg, Brewer, Cote, and Striegel use both methods for polysaccharide characterization and conclude that HDC coupled with multi-angle light scattering achieves more accurate molar mass distribution when compared to offline mols than SEC in significantly less time. This is largely due to SEC being a more destructive technique because of the pores in the column degrading the analyte during separation, which tends to impact the mass distribution. However, the main disadvantage of HDC is low resolution of analyte peaks, which makes SEC a more viable option when used with chemicals that are not easily degradable and where rapid elution is not important. HDC plays an especially important role in the field of microfluidics. The first successful apparatus for HDC on a chip system was proposed by Chimela in 2002. Their design was able to achieve separations using an 80 mm long channel on the timescale of 3 minutes for particles with diameters ranging from 26 to 110 nanometers, but the authors expressed a need to improve the retention and dispersion parameters. In a 2010 publication by Jalema, Markestein, Westerweel, and Verpoort, implementing HDC with a recirculating bidirectional flow resulted in high-resolution, size-based separation with only a 3 mm long channel. Having such a short channel and high resolution was viewed as especially impressive considering that previous studies used channels that were 80 mm in length. For a biological application, in 2007, Ha, et al. Proposed a microfluidic sorting device based on HDC and gravity, which was useful for preventing potentially dangerous particles with diameter larger than 6 microns from entering the bloodstream when injecting contrast agents and ultrasounds. This study also made advances for environmental sustainability in microfluidics due to the lack of outside electronics driving the flow, which came as an advantage of using a gravity-based device. Two-dimensional chromatography. In some cases, the selectivity provided by the use of one column can be insufficient to provide resolution of analytes in complex samples. Two-dimensional chromatography aims to increase the resolution of these peaks by using a second column with different physico-chemical properties. Since the mechanism of retention on this new solid support is different from the first-dimensional separation, it can be possible to separate compounds by two-dimensional chromatography that are indistinguishable by one-dimensional chromatography. Furthermore, the separation on the second dimension occurs faster than the first dimension. An example of a TDC separation is where the sample is spotted at one corner of a square plate, developed, air-dried, then rotated by 90 degrees and usually redeveloped in a second solvent system. Two-dimensional chromatography can be applied to GC or LC separations. The heart-cutting approach selects a specific region of interest on the first dimension for separation, and the comprehensive approach uses all analytes in the second dimension separation. Simulated moving bed chromatography. The simulated moving bed technique is a variant of high-performance liquid chromatography. It is used to separate particles and/or chemical compounds that would be difficult or impossible to resolve otherwise. This increased separation is brought about by a valve and column arrangement that is used to lengthen the stationary phase indefinitely. In the moving bed technique of preparative chromatography, the feed entry and the analyte recovery are simultaneous and continuous, but because of practical difficulties with a continuously moving bed, simulated moving bed technique was proposed. In the simulated moving bed technique, instead of moving the bed, the sample inlet and the analyte exit positions are moved continuously, giving the impression of a moving bed. True moving bed chromatography is only a theoretical concept. Its simulation, SMBC, is achieved by the use of a multiplicity of columns in series and a complex valve arrangement. This valve arrangement provides for sample and solvent feed and analyte and waste takeoff at appropriate locations of any column, whereby it allows switching at regular intervals the sample entry in one direction, the solvent entry in the opposite direction, whilst changing the analyte and waste takeoff positions appropriately as well. Pyrolysis gas chromatography. Pyrolysis gas chromatography mass spectrometry is a method of chemical analysis in which the sample is heated to decomposition to produce smaller molecules that are separated by gas chromatography and detected using mass spectrometry. Pyrolysis is the thermal decomposition of materials in an inert atmosphere or a vacuum. The sample is put into direct contact with a platinum wire, or placed in a quartz sample tube, and rapidly heated to 600 to 1000 degrees Celsius. Depending on the application, even higher temperatures are used. 3 different heating techniques are used in actual pyrolyzers: isothermal furnace, inductive heating, and resistive heating using platinum filaments. Large molecules cleave at their weakest points and produce smaller, more volatile fragments. These fragments can be separated by gas chromatography. Pyrolysis GC chromatograms are typically complex because a wide range of different decomposition products is formed. The data can either be used as fingerprints to prove material identity, or the GC per mega Siemens data is used to identify individual fragments to obtain structural information. To increase the volatility of polar fragments, various methylating reagents can be added to a sample before pyrolysis. Besides the usage of dedicated pyrolyzers, pyrolysis GC of solid and liquid samples can be performed directly inside programmable temperature vaporizer injectors that provide quick heating and high maximum temperatures of 600 to 650 degrees Celsius. This is sufficient for some pyrolysis applications. The main advantage is that no dedicated instrument has to be purchased and pyrolysis can be performed as part of routine GC analysis. In this case, quartz GC inlet liners have to be used. Quantitative data can be acquired, and good results of derivatization inside the PTV injector are published as well. Fast protein liquid chromatography. Fast protein liquid chromatography is a form of liquid chromatography that is often used to analyze or purify mixtures of proteins. As in other forms of chromatography, separation is possible because the different components of a mixture have different affinities for two materials, a moving fluid and a porous solid. In FPLC, the mobile phase is an aqueous solution, or buffer. The buffer flow rate is controlled by a positive displacement pump and is normally kept constant, while the composition of the buffer can be varied by drawing fluids in different proportions from two or more external reservoirs. The stationary phase is a resin composed of beads, usually of cross-linked agarose, packed into a cylindrical glass or plastic column. FPLC resins are available in a wide range of bead sizes and surface ligands depending on the application. Countercurrent chromatography. Countercurrent chromatography is a type of liquid-liquid chromatography, where both the stationary and mobile phases are liquids and the liquid stationary phase is held stagnant by a strong centrifugal force. Hydrodynamic countercurrent chromatography, CCC. The operating principle of CCC instrument requires a column consisting of an open tube coiled around a bobbin. The bobbin is rotated in a double-axis gyratory motion, which causes a variable gravity field to act on the column during each rotation. This motion causes the column to see one partitioning step per revolution and components of the sample separate in the column due to their partitioning coefficient between the two immiscible liquid phases used. There are many types of CCC available today. These include HSCCC and HPCCC. HPCCC is the latest and best performing version of the instrumentation available currently. Centrifugal partition chromatography, CPC. In the CPC instrument, the column consists of a series of cells interconnected by ducts attached to a rotor. This rotor rotates on its central axis, creating the centrifugal field necessary to hold the stationary phase in place. The separation process in CPC is governed solely by the partitioning of solutes between the stationary and mobile phases, which mechanism can be easily described using the partition coefficients of solutes. CPC instruments are commercially available for laboratory, pilot, and industrial scale separations with different sizes of columns ranging from some 10 milliliters to 10 liters in volume. Periodic counter-current chromatography. In contrast to counter-current chromatography, periodic counter-current chromatography uses a solid stationary phase and only a liquid mobile phase. It thus is much more similar to conventional affinity chromatography than to counter-current chromatography. PCC uses multiple columns, which during the loading phase are connected in line. This mode allows for overloading the first column in this series without losing product, which already breaks through the column before the resin is fully saturated. The breakthrough product is captured on the subsequent column. In a next step, the columns are disconnected from one another. The first column is washed and eluted, while the other column are still being loaded. Once the first column is re-equilibrated, it is reintroduced to the loading stream, but as last column. The process then continues in a cyclic fashion. Chiral chromatography. Chiral chromatography involves the separation of stereoisomers. In the case of enantiomers, these have no chemical or physical differences apart from being three-dimensional mirror images. To enable chiral separations to take place, either the mobile phase or the stationary phase must themselves be made chiral, giving differing affinities between the analytes. Chiral chromatography HPLC columns in both normal and reversed phase are commercially available. Conventional chromatography are incapable of separating racemic mixtures of enantiomers. However, in some cases non-racemic mixtures of enantiomers may be separated unexpectedly by conventional liquid chromatography. Aqueous normal phase chromatography. Aqueous normal phase chromatography is characterized by the elution behavior of classical normal phase mode in which water is one of the mobile phase solvent system components. It is distinguished from hydrophilic interaction liquid chromatography in that the retention mechanism is due to adsorption rather than partitioning. Applications. Chromatography is used in many fields including the pharmaceutical industry, the food and beverage industry, the chemical industry, forensic science, environment analysis, and hospitals.